total soluble Pi content is higher in shoot and less in root. We studied the expression of phosphate uptake transporters, PHT18 family, through QRT-PCR, and we found that at highest concentration, 20 millimolar or 10 millimolar, most of the member got repressed in their expression. At the same time, the other transporter PHO1, which is responsible for mobilization of phosphorus from root to shoot, its expression is increased even on 5, 10, and 20. So that's why it might be possible that due to the repressed expression of uptake phosphorus, uptake transporter, plants cannot take phosphorus from the medium, but at the same time, it keeps on mobilizing from root to shoot, and that's why it's creating a temporary phosphate starvation kind of condition in root and more phosphate in shoot. It was earlier reported that under minus phosphorus condition, the root apical meristem is completely exhausted. So this is important to study the meristematic status in our condition, and that's why we studied the root meristematic activity using the cyclin B1 promoter fused to a UIDA gene which encodes Gus protein, and every time when meristematic cell divides, it expresses. And we found that, as usual, it was reported there is no Gus activity under minus phosphorus. As soon as phosphorus available, it shows the expression, and after 5, at 10 and 20, it's completely abolished. So we can conclude that yes, excess phosphorus condition represses the meristematic activity, and thereby decreasing the primary root length, because primary root length is dependent upon the meristematic activity. The next question is the developmental program of the meristematic activity, because it was reported that in minus P, this is a determinate developmental program. Determinate means it cannot be reversed, it's permanent. The meristem is exhausted permanently. To know that developmental program, we transferred our plants which were grown at 20 millimolar and the plants which were grown at minus P again into the normal medium, which is our control medium, 1.25 millimolar, and let them grow for next 7 days. After 7 days, we measured their primary root length, and we found that the plants which were grown in 20 millimolar again starts growing, and their root length just doubled. And the plants which were grown in minus P didn't grow significantly. So we conclude that yes, it represses the meristematic activity. Excess phosphate represses the meristematic activity, but developmental program is indeterminate in nature and can be reversed when we put these plants into the control medium. There was earlier report that phosphate starvation causes accumulation of sugar in plants. So to check that, we did starch staining by simple iodine staining, and we see that there is a comparable phenotype of 20 millimolar and minus P condition. It was well documented that in minus P condition, iron level goes very high. And also, it was reported that on high concentration of phosphorus, it causes metal deficiency phenotype in plants. So that's why we studied different essential element composition under our condition. And we see that as soon as phosphorus level goes high, the iron level goes down, the zinc level goes down. So we can conclude that there is a crosstalk between phosphorus and iron and zinc. While in the other essential metal like manganese and calcium, they first increase till 2.5 and then they start decreasing. Up to this point, we have some clue that ethylene is maybe playing some role. To get a better idea, to get more insight, we performed microarray studies using Affymetrix ST array chip, and we choose 3 concentration, -P and control 1.25 millimolar and P20 millimolar as a access condition. And we did total transcriptome profiling in root and shoot separately. And this is the principal component analysis, which is showing that root and shoot completely different across all the concentration, whether it is -P, P.5, P20. Second, the transcriptome profile related to -P is distinct to the transcriptome profile which belongs to P1.25 or P20. Third, there is a relative closeness between the transcriptome of P1.25 and P20. And it is very obvious that because there is only difference of the concentration, and in -P there is complete absence of one nutrient. So this Venn diagram is showing the differentially regulated genes under -P condition and P20 condition, and this is against the control condition 1.25 millimolar. And we can see there is a very high number of genes differentially expressed in root and shoot under -P condition. And it is fine because the complete absence of the one of the nutrient can impact more transcriptome. So this yellow color shows the genes which is uniquely differentially expressed, and this red color shows the genes which is interacting in both conditions. To get further resolution of this data, we distributed these genes into the different patterns. These are the yellow line shows the unique pattern, and these are the interactions. So this one shows the upregulation in P20, 0 shows there is no change in the -P, and this -1 shows the downregulation of under P20, and 0 shows there is no change in the -P. So that's why we distributed these genes, and we found that 11 genes were uniquely upregulated under excess phosphate condition. There is 8 gene which is interacting in both condition, P20 and -P both. There is 4 gene which is upregulated in P20 but downregulated in -P. This is for root. Same thing is for the shoot. There is 30 gene which is uniquely upregulated under P20, and there is 16 gene which is interacting in both condition in -P and P20. Let's have a look on these genes. So first take the shoot genes. We have found several genes related to ethylene signaling. Like first this ACS11, this is a aminocyclopropane 1 carboxylate synthesis. It takes part in the biosynthesis of ethylene. Ethylene responsive factor 12, ethylene responsive transcription factor 13, expansin, hypothetical protein, ethylene responsive factor 55, ethylene responsive factor 03. These genes clearly indicate that something happening with ethylene. Ethylene signal transduction is significantly upregulated during excess phosphate response. If we see the genes which is upregulated in root, the annexin, this is the protein, it's a channeling protein responsible for the cation transportation, and it's also involved in the lateral root development. The MLO14 protein is recently reported that its mutant of MLO14 cause aberrant root phenotype. Nicotiana synthase is produced polyamine, which is responsible for the transportation of iron and zinc. And since we got that under excess condition, there is a phosphate deficiency, there is iron deficiency. So that's why these genes are upregulated to maintain the iron level. Further, FRO5, this is also a gene which is responsible for reduction of ferric into the ferrous, because plant can take up only ferrous form. So let's see some genes which are involved in the interaction between both conditions. It also has a FRD3 which is involved in iron-iron transportation. One is flowering promoting factor. A report showing that the overexpression of flowering promoting factor causes attenuation of the primary root in rice seedlings. There is some peroxidase which is indicating that there is some oxidative stress is also happening in excess phosphate. And there are some pectin esterase inhibitor which is responsible for the remodeling of the cell wall. And again we got the ethylene responsive transcription factor. So overall, these genes indicate that yes, ethylene signaling is upregulated during the excess phosphate response specifically. We validate these microarray results using the QRT-PCR. We choose some iron deficiency responsive gene, ethylene responsive gene, and root development. And this is the expression level. To finally confirm that is really something happening with ethylene, or to give more evidences, we used ethylene insensitive and sensitive mutants. In ethylene signaling transduction, there are 5 receptors which recept ethylene signaling. So these mutants like ETR13 and IN4 are the mutant of those receptors. And these are the IN2T and IN3 IL1 are some component of which regulate ethylene signaling at the nucleus level, at the transcription level. And this CTR11 is the constitutive triple response which always produces a sensitive phenotype, whether ethylene is present or not, but it's always constitutive. So we use these ethylene insensitive, ETR13, IN4, and IN2T, and IN3 IL1. These are the ethylene insensitive mutants. And CTR11 is the ethylene sensitive mutants. And we grow them in our condition, like control condition is 1.25 millimolar and 20 millimolar. And we've seen that there is some rescue in the phenotype. These are the wild type. And we see there is a rescue even in the primary root length, in the lateral root length. In the overall growth, there is a rescue in the shoot area, in the primary root length. So it supports our finding that ethylene is playing a role in attenuation of the growth of the plants in excess phosphorus. Since we are focused on the primary root length, and we also have the evidence of the meristematic activity, which is completely abolished under excess phosphate condition, we need to really estimate the root apical meristem size, because that is main component that governs the primary root growth. So what is root apical meristem? Root apical meristem is the number of cortical cells from quiescent center, the cell which is close to quiescent center, and the first elongated cell. So these number of cells from this region represents root apical meristem size. We measure the root apical meristem in wild type in our ethylene insensitive mutants, and we see that in wild type root apical meristem significantly decreased, and in ethylene insensitive IN2T mutant it rescued. In other different mutants like ETR13, we see that under control condition and in 20 millimolar it's increased. So it's a significant rescue is going on. In IN4 it also rescued. In IN3 EIL1 same thing, we got a rescued phenotype of root apical meristem. In CTR11, because it is a sensitive mutant, the root apical meristem decreased, and it is as expected. So on the basis of these all results, we proposed a common minimum consensus model that excess phosphate triggers the ethylene signaling, and ethylene signaling deregulated cell proliferation and thereby decreasing or attenuating the primary root growth. So conclusion, this study provides a comprehensive reference data set of the plant responses under excess as well as deficient phosphate regime. Excess phosphate supply indeterminately attenuates primary root developmental program. The negative growth of primary root is due to a decrease in the number of cells in root meristem. The transcriptional cues and morphometric traits indicate that excess phosphate response may be mediated by ethylene and signaling. This study not only helps in dissecting the mechanism of plant responses to excess phosphate, but also paves the way to identify the pathway and additional novel genes which can be utilized in phosphorus homeostasis and accumulation. Future prospects. So there is a need to functionally characterize all these genes like ethylene responsive factor, defensin, and amylo-like protein to know their exact role in phosphorus homeostasis and accumulation. Acknowledgements, USDA ARS grant. Dr. Sivan Sahi and Dr. Claire Reinhardt is a part of this work. Dr. K.G. Ragothama from Purdue University provided us for genetic material, cyclin B1 line. Dr. Rishikesh Bhalerao, Umia University student, provided the ethylene insensitive and sensitive mutant. Dr. Eric Skeller, Dartmouth College, USA, provided the double mutant IN3 EIL1. Dr. Ajay Jain, NRCPB, also provided the genetic material. Some mutants we got from ABRC, Ohio, and Mrs. Savine Weigel and Dr. Eric Rochika helps or hybridize the chips, microarray chips over the University of Kentucky, University of Louisville, and partial funding from Cabrin grant. Dr. John Andersland providing for his microscopy facility and everything, liquid nitrogen, so many things. And last, Biotechnology Center for providing the instrumentation facility. Thank you.